Good morning, everyone. Thank you for participating in the NIH Metabolomics Interest Group webinar series. Today's webinar from Drs. Ella Temprosa, Stephen Moore, and Evie Matei will focus on COMETS Analytics, a platform for consortium-based metabolomics analyses. Before introducing our speakers, I will quickly review a few logistics for you. Should you experience technical difficulties, you can contact us through the questions box in the expanded control panel or by phone or email. If you need to view live closed captioning, please click on the link that will appear in the chat box. At the end of the presentation, we will allow time for questions. If at any time during the presentation you have a question, please type it into the question box and we will ask it on your behalf during the Q&A session. So I'd now like to introduce our presenters for today. Dr. Ella Temprosa is an assistant research professor at the Department of Epidemiology and Biostatistics at the Milken Institute School of Public Health at George Washington University. Her research has focused on Phase 3 NIH-sponsored multi-center clinical trials for more than 20 years at the Biostatistics Center and a research center at George Washington University. She is the principal investigator at the Biostatistics Research and Coordinating Center for the NIDDK-sponsored landmark study, Diabetes Prevention Program and its Outcome Study. She has multiple leadership roles in COMETS. She's vice chair of the consortium, chair of the data harmonization group, and one of the principal architects of COMETS Analytics. Dr. Stephen Moore is an Earl Stadman investigator in the metabolic epidemiology branch of the National Cancer Institute. Dr. Moore's research examines physical activity, obesity, and diet, and their relationship with cancer risk. His recent work has focused on metabolomics, using its tool for dietary biomarker discovery and for uncovering molecular mediators of the association of obesity with cancer risk. Dr. Moore was a founding chair of COMETS, which is a consortium of 50-plus prospective cohorts that uses metabolomics to investigate the biology underlying major chronic diseases like cardiovascular disease and cancer. And Dr. Evie Matei is an assistant professor in the Biomedical Informatics Department at The Ohio State University. Her current research interests are to define disease-specific molecular characteristics using primarily metabolomics and genomics techniques to guide the search of candidate biomarkers and therapeutic targets for the diagnosis, prognosis, and treatment of disease, including cancer. To accomplish these goals, her lab is developing user-friendly methods to handle and integrate high-throughput omics data sets. So we'd like to start today's presentation of COMETS Analytics, a platform for consortium-based metabolomics analytics, with Dr. Ella Temprosa.
Thank you, Krista. Thank you all for your interest in COMETS Analytics, a reproducible and efficient platform for conducting consortium-based metabolomics research. Our presentation today is divided into 3 parts. I will provide an overview of COMETS and the need for COMETS Analytics and the guiding principles in the design of COMETS Analytics. Steve will give you a walkthrough using a demo, and Evie will provide a deeper look on what magic lies under the hood. So in the next few minutes, I will describe to you the principle that motivated the design and infrastructure, as well as the workflow that each analysis cycle will go through. We hope to convince you that this new paradigm in conducting consortium research has great potential to meet COMETS goals. So the mission of COMETS is to facilitate an open exchange of ideas, knowledge, and results to accelerate the study of metabolomics and its link with disease. Cohort eligibility consists of 4 criteria: more than 100 participants with blood metabolomics data. We require identified metabolites and platforms, either mass spec or NMR. We also require that there be follow-up for outcomes, for example, diabetes, cancer, CVD, mortality. There are also 4 membership types: steering member, associate member, affiliate, non-affiliate, and this depends on your affiliation and your expertise. All this information is available on our website noted below. So at present, the International Consortium has 53 cohorts. We have coverage of about 137,000 plus subjects and 126 researchers. 3 times a year, we have a call for proposals where investigators internally or externally can propose projects that will go on a steering committee review. At present, there are 10 projects ongoing, and here's an example of some of them. So the question then becomes, how do we conduct these analyses? We have 53 cohorts, many investigators. How can we move projects forward? This was the major goal of COMETS is to produce this coordinated multi-cohort high-impact publications to describe the relationship between metabolites and disease and other biological constructs such as aging and genetics. Many of you have participated in various consortiums where participant-level data are sent centrally. Challenges in that model include the dreaded data use agreement, central data harmonization by folks who do not have knowledge of the data collected, possible data stagnation over time, and the biggest problem really is the harmonization of metabolites across platforms and samples. At the outset, the steering committee of COMETS decided on no patient-level data sharing. So each data set or each cohort will keep their own data set. So we had to find another model. The COMETS Data Harmonization Group, which I chair, was given the challenge of figuring out how to conduct the data-related activities of COMETS. So our challenge was to provide an infrastructure to facilitate COMETS goals and coordinate data analysis efforts. And our vision was to create a platform that is disseminated and cost efficient. So here's the website for COMETS Analytics. It's open to the public with registration. So here are the guiding principles in developing the app. First, data autonomy was number 1. The data stays within the cohort, but we have centralized code to conduct analysis just by the user signing up for COMETS Analytics, and this improves security and monitoring. We require that using user interface and user experience design principles, we want to make COMETS Analytics simple and easy to understand. We have also provided interactive controls for user exploration of analysis results, and we want to make sure that it's efficient because we want to have minimal burden on cohort resources by running multiple projects at the same time, essentially batching the analysis. COMETS Analytics was also designed to be scalable and sustainable. The application lives in the cloud where we can monitor usage and load and adjust resources to meet longer-term needs and greater needs. The analysis we conduct in COMETS Analytics support reproducibility by using common code and metadata, which all reside on GitHub, which is now public. The templates are also available to the public so that anybody who would like to conduct exactly the same analysis that are being published or shown can do their analysis on their own cohort, for example, for validation. Here's the tool set that we have for COMETS Analytics. And here's the cycle for each wave of analysis. There's data preparation, metabolite harmonization, data model and integrity, then we conduct analysis, and finally the meta-analysis. And I will go over this with you in detail. So in metabolite harmonization, our purpose is to maintain an up-to-date master list of metabolites across platforms and cohorts to establish a universal metabolite ID. Obviously, each cohort and each platform have their own metabolite list, and the goal is to make sure that using some chemical identifiers, for example, HMDB, KEGS, that we are able to say that this given metabolite from this cohort is this certain metabolite from this other cohort. And we employ, we also include the mass charge ratio and the retention time information in the metadata. This spreadsheet or this metabolite harmonization file is available to the public on the COMETS Analytics website. We also are able to classify metabolites using data from the Metabolomics Workbench and Metabolon. Here's some statistics on metabolite harmonization status. So we started with about 3,000 metabolites at the outset and have progressively increased over time with a current total of about 23,000 metabolites harmonized. This is the distribution of metabolites across the number of studies. So we have 2062 metabolites harmonized across at least 2 studies right here. And here at the lower end, 7 metabolites are available in all 18 studies that have been included in some of the projects. So at present, we've found that we're increasingly successful in harmonizing metabolites so that in recent runs, more than 90% of metabolites were able to be harmonized for cohorts. So here's the metabolites by platform. A majority of metabolites come from Metabolon, next is BrainShake, the Broad, West Coast Metabolomics Center, Biocrates, and the LEAS NMR. And here's the overlap. So there's 121 metabolites that are both in the Broad Institute and Metabolon. So as we harmonize more cohort metabolites, we expect these numbers to increase. What we found is that the lipid is the overwhelming class of metabolites that we have in COMETS. Next is xenobiotics, amino acid, and all the way down to energy. In data preparation, this is the key in running the analysis. Each cohort is required to assemble their own data into standard Excel format from a variety of different sources from Excel, Stata, SAS, or R, and this file is then used for harmonization and analysis. We, in this Excel file, which Evie and Steve will go over in more detail, we require 5 worksheet tabs: metabolites, which is a metadata, subject metabolites, subject data, a var map, which maps data that we require for analysis to the cohort variables, and models. Here's an example of the tool that allows you to create these Excel files that you need for analysis. In terms of data and model integrity, we need to validate the data and models before running analysis. Steve calls this the smart pre-analytics before conducting analysis. As you can imagine, if you run models on different data sets, you might have to do some adjustments in terms of availability of data, variances among variables, and specification of stratification. So, for example, if the cohort is only only females, then you would remove stratification by sex in your analysis. And this is done, handled automatically by COMETS Analytics. In terms of, after it has passed all the checks and rechecks of both data and models, we then proceed to cohort analysis. So this is to conduct the cohort analysis. We want to make sure that we're conducting this in a reproducible and valid way. We have 3 modes of analysis: interactive, batch, and super batch, which Steve and Evie will go over. There's a tutorial to guide the analysis for people who are newbies. And in the interactive mode, we allow interactive controls that allow you to explore the analysis results. The analytic modules are available for correlation and hierarchical clustering and linear models at this point. Here's a picture of the COMETS tutorial that we've recently completed. It allows you a step-by-step view of the analysis, also to describe the different details about running your own cohort analysis. Here's an example of a screenshot of correlation results where you can do some interactive metabolite tagging, which Steve will go over, and also filter the results of the table. Here's an example of a correlation heat map that you get from interactive mode. Steve will go over this in more detail, but it gives you a sense that, you know, models can be specified with a point and click in the user interface. Finally, the meta-analysis allows us to synthesize and aggregate the results from the different cohorts. The main benefit of using COMETS analytics is that there is standard output format for each cohort. We ask that cohorts send these aggregated results centrally, so no patient-level data. We have random or fixed cohort effects that we apply in the meta-analysis. And after publication, the public are able to run the same analysis on their data for their own validation using the templates that we provide on the website. So in summary, COMETS Analytics provides a new paradigm in conducting consortium-based research that promotes reproducibility, scalability, sustainability, and validity to advance our understanding of the metabolomic basis of diseases. So here's some of the websites that I talked about. And next, I pass it over to Steve to give you a demo.
All right. Krista, can you hear me?
Yes, we can hear you fine, Steve.
Okay, excellent. All right, so before I start the demo, I have 3 slides to present a bit of context to help you understand what's going on with the demo. So in COMETS, many of the cohorts cannot send their data to a centralized repository, and that means that we have to perform analyses on a disseminated basis. And what that means is that each institute runs its own analysis, and then they send the results centrally for meta-analysis.
Steve, we can't see your slides right now. Could you just pause for 1 second while Stephanie figures out? Can you share your screen, please?
Yes.
We'll tell you when we can see them. Yes, we can see them now. Great,
all right.
Ready to go.
Excellent. So 3 slides. So each institute runs its own analysis and sends results centrally for the meta-analysis. But that approach can be challenging with complex data like metabolomics. COMETS Analytics is designed to facilitate the analyses in 3 specific ways. So first is enhanced quality control. COMETS Analytics has built-in checks and nudges to help investigators provide high-quality data that's in a standardized format. Second, it's simple to use. It doesn't require learning any specific programming languages, which should help ensure broader participation. Third, the data are output in a meta-analysis ready format, which should help to accelerate the analyses and prevent confusion on the back end. And as I walk through the demo, I'm going to point out some of these features as I go. I also wanted to provide a very quick snapshot of one analysis done with COMETS Analytics, and this would be for age and metabolites. And so far, 19 cohorts have completed the analysis for this project. It's one of the COMETS proposals. Overall, we have in at least 3 studies 1,715 metabolites. The maximum size would be 27,000 people, and that's for the 7 metabolites that Ella noted were present in all 18 of the cohorts, actually 19 now. But we also have 460 metabolites with at least 10,000 people, and we have an N of greater than 5,000 for 1,300 metabolites. So the coverage is very deep. And this last slide just presents a visual of the random effects meta-analysis so far. So based on this, there's hundreds of different Bonferroni significant associations. I've separated them by chemical class here, but this is just to give you an idea of the flavor of what the final result may look like. So with that said, I'm going to now move on to the demo. This is the login screen for COMETS Analytics. It's at cometsanalytics.org. We have two different ways to log in, either your Google or Facebook identification, or you can actually sign up for your own separate login. And so I'm going to use my Google login for this. Oh, hold on. All right. So this is the welcome screen. It provides an introduction to COMETS Analytics. We've kept it very simple for now, but in the future, this will include a tutorial, possibly a video archive of this presentation, and a frequently asked questions section. Now, at the current time, COMETS Analytics has one primary function, which is to produce correlational analyses. Later this year, we'll be adding logistic regression and other functions. The correlational analyses can be found at this tab here. And to help people get familiar with COMETS Analytics, and this is important, we've included a sample input file that you can download right here at this button. It's simulated based on a large data set with a relatively good span of metabolites, 611 metabolites. And the results that you get from this are pretty similar to real data. So we prepped this file so that users can test drive COMETS Analytics, get familiar with it, without having to go through the trouble of creating their own data input file first. And it also provides a template that the analyst can refer to when setting up their own data set. So I'm going to open this, show you a few of the inputs. The first two tabs, the metabolites and subject metabolites, are here. And these provide metabolite metadata and the levels of metabolites in the study participants. So in the first tab, the rows are metabolites, and the columns are different information about those metabolites, different items of metadata, like their pathways. The second tab is the subject metabolites, and here the rows are participants, and the columns are the different metabolites, and then obviously the cells here are the different values of those metabolites for those participants. So this particular setup is one of our first nudges toward data quality. So typically, epidemiologists are analyzing their data in a single flat file like this one here, and they don't provide any metadata about the metabolites. So by requiring them to provide that metadata, it makes it possible to link the metabolites from one study to another. One of the catches with metabolomics data right now is that metabolite names aren't yet standardized. And so providing additional metadata, for instance, an alternative name that's in English language instead of programming language, some information about the super pathways, maybe some unique identifiers from online databases, it makes it easier to link metabolites across cohorts. The third tab includes the participant covariate data. And this tab is relatively straightforward. You have the participants and then things like age, their gender, are they female, smoking group, and so on. That's relatively straightforward. The fourth and fifth tabs, the VAR map and the models, are used to ensure coordinated analyses. So Evie will go over these in more detail, but in essence, these tabs lay out the details of the analysis. The VAR map tab, for instance, shows how to code and name all of the different covariates in the study. And the models tab details the different models for analysis. So each one of these rows is a different model. For this demo, I'm going to make one quick modification so that this will run a little bit quicker. Hang tight just a sec. All right, save that. And flip. All right, so now back to the COMETS Analytics screen. On the first step, after selecting your cohort, that is, all right, and this is the file that I just downloaded. The first step is to check the integrity. And this is very basic checks. It ensures that the metabolite names and covariate names match between the different tabs. It checks the metabolite metadata against our master list of metabolite names on the back end also to see if there's any matches or other information that we have that may be relevant. And then once this is complete, you can perform the analysis. And so that's the next step here. So if the input tables are set up properly, then what we're asking the consortium members to do is very simple in a sort of blink and you miss it kind of way. We literally want them to just say, click this batch button, choose all models, enter their email address, and click run model. And after that, we get this pop-up that says, your job is sent to the queuing system for processing. It's just letting you know that your analysis is in a queue and that we'll send an email when all those results are done. On average, for this particular data set, which has 1,000 people and 600 metabolites, it takes about 5 minutes. And so we're going to check up on that in about 5 minutes. But in the meantime, there's a lot happening behind the scenes for this analysis. And so I'm going to walk you through some of those steps using the interactive user input mode. And this is a different way of doing the analysis. It's a point-and-click interface that allows you to run one single model at a time. So the first model in our analysis is an analysis of age and its association with all metabolites. And it's adjusted for gender. All right, in this case, it's the variable female. So I can enter that in the interactive mode here, click Run Model, and it's going to run. It takes about 30 seconds. Now, in the meantime, let me show you what's going on in terms of the spreadsheet. All right, so in the spreadsheet, we have this Models tab, and the very first model is the gender-adjusted analysis, all metabolites, age, and adjusted for females. And so what I'm showing you now is that I just ran the same model that we specified in the spreadsheet and that we can run in a batch mode. I'm showing that we can also do that in interactive mode through the point-and-click interface. And so going back now to the COMETS Analytics website, you can see the results for this particular analysis. Typically, let me add one more thing real quick. So Ella mentioned that the COMETS Analytics program has some smart analysis functionality, and this message here on top is an example of that. So it says one of your models specifies female as an adjustment value, but that variable only has one possible value. Model will run without female as an adjustment. What COMETS Analytics has picked up in this case is that everybody in this cohort is female, 100% of the participants, and therefore adjusting for that covariate doesn't make sense. So it actually adjusts on the fly the model so that it runs a model that actually gives valid output, ignoring that female variable. Okay, so results are below, and typically I would start by sorting by p-value. And we can see lots of statistically significant associations here. The metabolite on the top, vanilla mandolate, or VMA, is an interesting metabolite that has some biology related to neurochemical responses. Some of these other metabolites have come up in other contexts. This is citrus. Down a little bit lower, we have a metabolite, C-glycosyltryptophan, which has been found before in previous studies. So lots of interesting hits. You can see that there's multiple pages of this. If you want to roll through all of the analyses, you can just click from page to page. But also, you can download the results into a single Excel file, and that's what's at this button here. So if I click this, and give it about 1 second, we see the same set of results fully downloaded in Excel, which may be easier for some people to work with. And so again, if I sort by p-value, I have VMA on the top, same correlation, same p-value. So it's the same set of information as what you saw on that prior screen, sometimes with a little bit of additional metadata about those metabolites. Some of this is merged in from the back end. So this is letting you know that, for instance, the variable that was entered as vanilla mandolate underscore VMA underscore has a proper name of vanilla mandolate parentheses VMA, the English language equivalent. All right. In addition to the interactive mode, we have a batch mode. And that's what I started with the all models. But in addition to that, you can run things one at a time. And so that's what I'm doing now. And the first model, the gender-adjusted model, is what I just ran in interactive mode. It's accessing that model through the spreadsheet again. Anything in the spreadsheet is passed on to that batch dropdown, and it knows enough to know that that's one of the models you may want to run, and it pre-populates it, so you can just select it automatically. Now, let's see. Yes, one more thing. So each of these rows is a different model, and each row will produce a different table. And so the model I've been running is just adjusted for female status, but this model here will basically be identical to the first one, except additionally adjusted for smoking and BMI, race, education, alcohol, multivitamin use, hormone use, and so on. Some of the other models actually include stratifications. So for instance, this model is stratified by BMI group. So it's going to produce 611 metabolite age correlations times the 3 different BMI groups in our study. So you can add on to these different models with stratifications, with where statements. It has pretty much the full set of functionality that you might have in, say, a code, a programming language. And again, that interactive mode that I just ran, that batch mode that I just ran, produces BMA at the top. Okay, so circling back to the start, we selected all models at the beginning, and it basically did essentially the same thing as if we had just run all of these different models individually. And so it's time to visit my email. And there is a caveat here. I said 5 minutes, but if someone else is using the system now, it could take a little bit longer. It's a live system. But here we have it right at the top. Okay. The email itself includes a link to the zip file, which I'll show you in a second. It also includes messages with details about the analysis itself. So first, for instance, it took about 4 minutes. Then for the individual models, it has some warnings, but these are really just specific details. So, again, with the gender-adjusted model, it has the one of your models specifies female as an adjustment, but, we're going to run it without since all of them are female. So this is a way of documenting the different decisions that COMETS Analytics has made about the models right in the email itself so that they can't be missed. Moving to the zip file, we can now see all of the different models that were run. So the gender adjusted, if I clicked on that, you'd still see the BMA on the top result. The BMI stratified, for instance, will have 3 different sets of results. Let me even just show you. One for each different BMI group. And so you see here the strata variable at the end. This is strata 3. The N for that strata was 269. If you scroll down far enough, you get different strata. So this would be strata 1. I believe that's the normal weight group, had an N of 351, and these are the correlations for that group. So the zip file packages all of those different things together. This is actually 2 different analyses in one that's related to age and metabolites, another that's related to BMI and metabolites. But even beyond that, it includes a little bit of additional information to help the investigators with their meta-analysis. And so this file, the descriptive file, for instance, includes basic descriptive characteristics on the participants in the cohort. So again, for female, we had a value of 1 for N,000 people. That was 100% of the participants in this study. And these proportions tell you the proportions for different smoking groups, BMI groups, and so on. We also provide some output for continuous variables as well, including for all of the metabolites, just so that the user has that basic descriptive information for any given cohort as a way of helping to identify or perform any kind of troubleshooting. Beyond that, there is also a file showing the original input minus any of the study-specific data. The whole point here is that we can't move the study-specific data around, But we can move, for instance, the metabolite metadata, the variable map that they used, as well as the models. So this just shows the metabolite metadata from the study. This shows the variable map that they used, which could be useful if you're investigating some kind of error or problem with the study. You can check to make sure they use the same variable map, and the models that they used here. So with that, I've come to the end of this part of the demo. I think it's actually time to move on to Abby.
All right, can everybody hear me and see my screen?
Yes, Abby, we can hear you fine and see your screen.
Okay, great. So I want to talk a little bit about what goes on behind the scenes and a little bit more about formatting, because this is going to be important as people use the software. So as a brief review, the input of the data is an Excel spreadsheet with 5 sheets. The first one is the metabolites that has the meta information for metabolites. The second one is a subject metabolites that has the abundance data. So here the metabolites are columns and the samples are rows. The subject data sheet has the meta information related to the subjects. The VARMAT sheet has the variable mapping that is important in terms of the software, and we'll get into detail about that particular sheet. And then finally, the models. So again, this is basically what your spreadsheet looks like, and Steve has gone over the metabolites, the subject metabolite, and the subject data quite a bit. So here we're going to focus on the VARMAP sheet. So the VARMAP sheet looks like this, and I want you to focus here on the first column. So this first column we called VAR reference, and this is a variable name that's going to be used across cohorts. So if you're involved in a meta-analysis, this is going to be predefined. So for example, if you have the ID here, this ID pertains to the cohort subject ID, and in another study, it's also going to be called ID. The second column, VAR definition, just has a description of that particular entry. And then the third and fourth column are what you call the variable. So this cohort variable here, it's in your data, the ID, which is used across all cohorts, you're actually calling sample ID. And this just avoids you to have to recode all of your variables. So you can just do the mapping directly using this VARMAP sheet. And then here, we're also requiring users to input the type of the variable. So the 2 possible entries are continuous and categorical. And this is particularly important for the smart pre-analytics that the software has, because it will check whether the data is continuous. It will check whether the data is categorical. And for those that have a little bit of experience with our programming, you know that factors can sometimes be an issue, and so we want to check and make sure that the data is in the proper format for the analysis. And then finally, if you have any additional notes that you want to include, you can include those. And this is particularly important for note keeping, but this will not affect any of the analyses whatsoever. So again, the important things to remember here is that VAR reference is what those variables are called globally by all the cohorts, and cohort variable is what you call for your own data set. So the model sheet looks like this, and Steve has gone over this, so I won't spend much time other than to say that the variables that are included here must be the VAR reference variables. So those are the ones that are called across all the cohorts. So don't use your local variables, use the global variables here. And sorry, let me just, oh, no, sorry, okay. So in order to put all together this data in the Excel spreadsheet, we do have this sample input data, and you can download it to look at it. That's definitely helpful. And then we also have a little utility here, create input, where you can input CSV files one by one to help you create this input file. And once you have this input file created, then all the analyses run smoothly. But you do have to remember some constraints. So some of the constraints are that whatever samples names are used in your subject metabolites sheet that contains the abundance data, they have to match the metadata for the subject, so the names that you use there. Same thing for the metabolites. If you have metabolites in the abundance data, those metabolite names must match the names that you include in the abundance data. And then for cohort variables, you have to be sure in the var map sheet that whatever variables you map actually exist in your data. So to help out the users, we've developed extensive data integrity checks. And again, this is the smart pre-analytics features of the software. And if there's anything wrong with your data, it will return an error. And an error means that the program stops. It will not do the correlations until you've fixed everything. But we try to return meaningful errors, meaning you'll know exactly where to go in your Excel sheet and what you need to fix. So some examples of errors are the software can't find appropriate variables. Some of the metabolite or samples have missing or non-matching metadata. The adjustment and stratification or exposure and stratification variables are the same because then you're not going to be able to run your models. So some examples of an error is here. For example, here the user input ID in the cohort variable column of the VARMAT sheet is not found in the subject data. So whatever label you have put in your data for the ID isn't found in the abundance data, so you have to go and fix it. Please email us. So if you end up using COMETS Analytics and you get an error and you have no idea what it is, let us know. That's how we're able to improve the software and make it as smart as possible. The other checks that we perform are at the model level. And here, those are mainly warnings. There's a few errors, but most of them are warnings. And warnings, the difference between a warning and an error is that a warning will not stop the analysis, whereas an error will. So some examples of warnings is the check that adjustment variables have at least 2 unique values. So Steve had an example of that with the adjustment being female because there was only 1 value. It threw a warning and it automatically dropped the variable. Other checks include the check that all covariate have non-zero variance. Again, if that check is violated, then those variables are dropped. It also checks for correlated covariates and for linear dependencies, and it'll automatically adjust the model. So here again, if you get some type of warning that's not meaningful, you don't really understand what it is, let us know and we will adjust. We've spent a considerable amount of time doing these data and model integrity checks, and as different scenarios come up, we want to continuously update them. Another aspect of COMETS Analytics that is very important is the harmonization. And this will check whether your metabolite ID is present in all the other COMETS cohorts and if they can be harmonized. So if it does find a match, then it will return the harmonized ID. If it does not find the match, then it will just return your name. So this will not affect any of the downstream analyses, but it does make a difference in terms of the meta-analysis if you're involved in a large meta project. So as I mentioned, the analysis will proceed regardless of whether metabolites are harmonized, and the harmonization just happens automatically. And it will return to you the total number of metabolites in your analysis, the total number of metabolites that are harmonized, and the total number of metabolites that are non-harmonized. And as Steve mentioned, it's important to download this list and send it to the harmonization group so that we can expand how many metabolites are harmonized across cohorts. So for those of you that do not want to use the COMETS analytic web interface, although it's great, it's a nice GUI and it works really nicely, if there are any privacy issues, for example, that with your cohort, you can run all the analyses locally. And we have an R package, and it's publicly available in GitHub, and the address is here. And the package includes a detailed vignette, and I have a snapshot here on the right. And it gives you a brief workflow of all the functions that are present in the R package. These functions are the ones that are called by the GUI. So whether you use the GUI or you use the R package, you will get the exact same results. And we are also working on creating additional vignettes for specific meta-analysis cohorts and specific models that need to be run. A little bit more about the interactive versus batch processing. So in the interactive mode, you can define on the fly what model parameters you want, either through the GUI or through a local R console. And here you're only running one model at a time. In batch mode, you're also running one model at a time, but you're calling one of the models that you have already defined in your model sheet. I apologize, this is a mistake here. I'll fix that. It shouldn't be the var map sheet, but it should be the model sheet. So basically, it's just so that if you have all models that are predefined in your models, you can actually just give the name of that model, and it'll fill in all the parameters that are in there, which is useful if you have a lot of covariates. Now, on top of that, there's a super batch mode. And this basically runs all the models that are present in your model tab. So this is assuming that you've already passed the data integrity check. But for each model, it'll also perform the model integrity checks, and then it'll create one Excel spreadsheet per model, and it'll zip everything up. And you'll have everything concerning the warnings, as Steve mentioned, the basic stats description of your cohort, your input data, and all relevant information concerning your data and for future, that will help you for doing your future meta-analysis. So here is the link to the COMETS Analytics software again. And right now we are at version 1.4. And the key features is that we have secure access through the user web app. And we also have the publicly local package available, our package available. And this web app, I should mention, allows multiple users at once. And so it'll automatically queue all the jobs on the back end and send you an email once the results are ready. The framework is very flexible, and that's partly due to the extensive model checks that have been implemented. So if you want, different adjustments, if you want stratified analyses, and it'll automatically check that there's no collinearities or anything problematic with your data, so the model can't be run. It'll automatically adjust your models and give you warnings. So this really allows for you to have reproducible analyses. And as people, are putting papers together, if you're diligent about putting together your model sheet, that'll help you a lot because you have everything recorded in a nice format. You can also perform the batch processing, and that facilitates meta-analyses. And we worked pretty hard as well with the documentation. So there's a nice tutorial. The user interface is also user-friendly. So hopefully it's very intuitive, and we have the R package vignettes. So the next version, we look forward to implementing generalized linear modeling and logistic regression, and we will also incorporate pathway analysis. And again, this is the address for the R package, which is publicly available. And one thing I should mention is if people are using, the R package and they have any trouble, then they should create issues directly through GitHub, because we do track those and we use those internally as we develop the software. So thank you for your attention. I hope you're excited about trying out COMETS Analytics. This is definitely a large group effort from the COMETS Harmonization Committee, the COMETS Tutorial Committee, CBIT, which is responsible for the web development, IMS, which is involved with the harmonization, and of course funding from DCG and DCCPS, which this project wouldn't happen without that. So we look forward to answering some questions and to hearing some feedback about the software. Thank you.
Okay, great. Thank you so much for sharing everything about COMETS Analytics with us. We did get one very quick question that I'm going to address immediately, and would the slides be distributed? What we actually do is we'll be posting the webinar on the NIH Metabolomics Scientific Interest Group webpage, especially for this one, since a large chunk of it was a tutorial. That's where it will go. So also, if you do have a question, please feel free to type it into the question answer box. So the next question is, you had mentioned that there were, for privacy issues, you could run it locally or in the GUI. And when people run it differently, the intent is still to send the data in for meta-analysis. Is it correct that regardless of whether you run it locally or in the GUI, even though it's exactly the same, are there any additional challenges from combining those data?
I can take this.
Go ahead.
No, there shouldn't be, because all the functions that are used from the web interface are exactly the same that you would run locally if you were to do it on the back end. So there should not be. There's no issues there, and the results will definitely be the same. The output formats will also be exactly the same, because the functions that are used to output the resulting, Excel sheets or zipped files, if you do the meta or the super batch mode, those are all functions that are part of the R package. So it will be identical for the meta, and it will not affect, the meta-analysis in any way.
Right. Instead of getting an email, you will just get it, created on your working directory in R.
Great. Thank you very much. The next question, does running analysis in batch mode make for easier meta-analyses than interactive mode?
I'll take this, and the answer essentially is yes. So if you ran identical models in interactive mode, then the results should be the same. But the main reason for running it in batch mode is the level of documentation. The program documents which model was run as it goes, and if it has that, one.age gender-adjusted label attached to it, we know exactly what that model is. Whereas if it's run in interactive mode, we'd actually have to look at the specific adjustments and kind of piece it together.
Also, the interactive mode is like, if you think about it, it's more exploratory. So we didn't show you, but there's this feature called metabolite tagging, where you run the analysis in interactive mode. You saw that some metabolites look interesting, and you want to save those metabolites for a deeper dive into relationships with other covariates. You can actually save those metabolites in the bin to say, these are metabolites. I want to look at these, tagged metabolites for the next batch of models. So you could think of it as more an exploratory tool, you know, with your data rather than, you know, using predefined or, you know, predefined model specifications that you usually specify in your analytic plan.
Thank you. Next question is, how do you deal with unknowns?
This is Steve, and I'll take the first cut at this answer, and Evie and Ella, if you have other feedback, please chip in. So if the unknown doesn't have a documented track record of being reproducible, we do not analyze it typically. So COMETS focuses on identified metabolites, and you know, most of the platforms that epidemiologists in our group have been using focus solely on identified metabolites. With that said, we have decided to allow the unknowns from Metabolon because they are reproducible. In other words, the metabolite they call X12603 is always the same in every study. And we know, for instance, that there's a specific gene that, you know, codes for it. We know that that it's related to sex steroid hormones, that it's higher in women, regardless of which study. But for unknowns that don't have a documented track record of reproducibility, we typically would not include those in our analyses.
Thanks, Steve. Does COMETS Analytics currently allow for to quantify metabolites from NMR data?
It should be, yes, the same, although because our initial analyses focus on correlations, the quantified aspect of it wouldn't necessarily come out. In other words, the correlation doesn't really have any component that tells you about the quantification. It's a little bit different than linear regression that way, where you can, you know, specify per unit increase in the metabolite what the exact effect would be on an outcome.
Thank you. Next question. I know you mentioned adding regression analyses capability soon. How about other statistical tools? Will there be updates when these statistical tools have been added?
We're constantly adding functionality. One of the biggest projects that we did this year is that smart pre-analytics, and that will allow us to have really good data for our different modeling that we will use. So Evie mentioned, you know, the logistic regression and generalized linear model where you identify, you know, the link distribution that will allow you to do Poisson and other regression models. So we will, you know, we will roll out as needed, you know, to serve the projects that have been approved. And if you have any other suggestions, you can certainly create an issue on our GitHub site. But the goal is, you know, to really focus on getting the projects that we need for getting the projects serviced for the consortium.
So and to add, sorry. To add to this, I think, you know, getting feedback is important because it helps us prioritize what will be rolled out next. So yes, I mean, this is a start, and I think this project is going to continuously expand.
Great, thank you. For those of you that need to jump off, please feel free to do so, but we have several more questions. And if our speakers are able to stay on for about 5 more minutes and answer them, we could get those answered for those individuals who asked them. As I said, if you would like to, it is the top of the hour. If you would like to leave the seminar, I just want to thank you very much for attending, but I will try to get these last few questions answered for those who asked them. The next question, how is imputation handled? Can we choose different imputation methods?
I'm going to turn that over to Ella and Evie.
Yeah, so this is, you know, the next batch of the next big project that we need to figure out is, you know, now that we've moved, we're moving away from correlation analysis, we need to consider all of these topics in our, I guess, you know, smart pre-analytics, including, you know, normalization, standardization, imputation. And so, if you have any ideas or some thoughts about this, we have, a lot of considerations, obviously, to think about, but, so it's still under development. So we welcome any discussion about this.
At the current time, we ask each cohort to perform the imputation on metabolites that they normally use, figuring that, each individual study knows best its own data. I think a large number of cohorts have been for values that are below the limit of detection. They're imputing either the lowest observed value or, the lowest observed value divided by 2 or some variant thereof, which is a reasonable method for imputation, although not necessarily the statistical optimum.
Okay, thank you. How do you account for matrix differences? Are these metabolites from serum or plasma or both? And what about urine?
This is Steve again. We restrict to blood, so plasma or serum. We're not including urine yet, although that's in the plans as well. With serum and plasma, typically we found that metabolite levels are highly correlated across those matrices. And so in rank-based analyses like the ones that we're doing with correlations, those factors don't tend to matter as much. In other words, we're not examining a nanomolar increase per, deciliter of blood or something like that. And so the comparability between serum and plasma doesn't quite matter as much without the absolute concentration. But we would, for instance, meta-analyze serum and plasma separately and examine whether there's heterogeneity across those. So we'd perform the overall analysis, then we'd stratify by sample type, look for heterogeneity, and if there was heterogeneity for certain metabolites, then we would dig in deeper into that.
Okay, this is an easy, quick one. You mentioned that to create the universal metabolite ID, you had help from IMS. Could you please say what IMS stands for?
Information Management Services. It's a consultant company.
Great, thank you. Could you elaborate on the metabolite harmonization process?
Right. So this is somewhat complex and a little bit iterative. Basically, each cohort, as they run their analyses, they pass through to us their metabolite metadata file. We ask them whenever possible to include all of the metadata that was provided by their lab, as well as any other information they may have added. And what we're looking for in particular are unique identifiers from online databases. So many of the different labs provide, for instance, HMDB IDs, PubChem IDs, ChemSpider. I think those are the 3 most common. And so by kind of chaining together those unique identifiers, you can link up metabolite names from cohort to cohort to cohort, and even to some extent populate, missing entries for different metabolites. Like if, it's HMDB is provided in one study and then another study provides HMDB and ChemSpider, then the HMDB and ChemSpider for the original metabolite too. So when I say it's an iterative process, that's in part what I mean is that we're kind of filling in information as we go by linking across these unique identifiers. For about 5% of metabolites, we are forced to go to hand searches. And every week I meet with IMS for about an hour and we talk through the latest exceptions of the week. We'll look at their names, we'll look at their INCHI keys, we'll compare them to online databases, and come up with a resolution based on a previous rubric. And that rubric is relatively complex. I'm not going to get into it now, but we do try to follow a set logical system for making this kind of calls.
Thanks, Steve. So thank you all for hanging in there. We have 2 more quick questions. I guess to set up the models to run the different cohorts, the COMETS analytics would need to work with the different cohorts to create the models and then let them run at the cohort level, correct?
Usually there's, so for a given project, there's a working group, and that working group kind of works together to identify the models that are needed to address the specific aims in their project. And so, some discussion on, what models will be used, what adjustments, what stratification to assess heterogeneity occur, at that level. And then those discussions get translated to the model worksheet, and that worksheet then gets sent to the cohorts that have opted into the project.
Right. So the key focus is that we have a standard set of models that all cohorts should be able to run. And when there's issues, COMETS Analytics is smart enough to get around that. So with the example of, a study that's all female, it knows that it should just drop gender from that model. And then the results can be more sensibly, it'll run, and the results can be more sensibly analyzed. But yes, the idea is that that whole tab of models is sent intact to each of the studies, and then COMETS Analytics works around the problems.
Thank you. This should be a very quick last question. Are the p-values in the output files the raw p-values or already adjusted for multiple testing?
They are raw. The ones reported are raw p-values.
Great.
Yeah, we find
Thank you. Oh, go ahead, Steve.
We find that that's generally what people want, and then they can apply their own correction.
Great. Thank you so much. So thank you for the enthusiasm and all the questions and staying on a little bit longer to allow everyone to get their questions answered. I want to thank you all for your attention and active participation in today's NIH Metabolomics Interest Group webinar. We would especially like to take this opportunity to thank our presenters, Drs. Ella Temprosa, Stephen Moore, and Evie Matei, for an excellent presentation. And we welcome your feedback to inform future webinars. So thank you very much, and everybody have a wonderful week.